For 115 years, the largest mosasaur skeleton in North America has been labeled Tylosaurus proriger. It hangs 3 stories above the floor of the University of Kansas Natural History Museum, coiled, almost 14 meters long, an estimated 8 tons in life. It is the official state marine fossil of Kansas, and in May of 2026, a doctoral student at the American Museum of Natural History published a paper that took the label off. The skeleton is not Tylosaurus proriger, it is Tylosaurus rex, and the reclassification is not a relabeling exercise. It is the result of a researcher noticing that the teeth on a museum collection specimen were too finely serrated, and following that observation through 12 skeletons across 4 institutions. This is the story of what hunted in Kansas 84 million years ago, how scientists learned what it ate, and why the king of that ocean spent a century hiding under the wrong name. To find the animal, the ocean has to come first. 84 million years ago, Kansas did not exist. Neither did most of the central United States. A warm, shallow inland sea called the Western Interior Seaway ran from the Gulf of Mexico to the Arctic Ocean, splitting North America into 2 land masses. The western half, where the Rocky Mountains were uplifting, was called Laramidia. The eastern half was called Appalachia. Between them, where Kansas, Nebraska, and the Dakotas now sit, was open water roughly 400 meters deep at its maximum, with surface temperatures between 16 and 22 degrees Celsius. The seafloor was periodically anoxic, which preserved any large skeleton that sank to it. The chalk that records this is called the Smoky Hill Chalk member of the Niobrara Formation. It outcrops across western Kansas in pale, eroded badlands, and it is made of microscopic plankton called coccolithophores that snowed down through the water column for millions of years. Embedded in that chalk, paleontologists have pulled out fish, sharks, sea turtles, pterosaurs, toothed birds, plesiosaurs, and mosasaurs. A mosasaur is not a dinosaur. It is a squamate, the same order as monitor lizards and snakes. Mosasaurs descended from land lizards that returned to the sea around 100 million years ago, and over the next 34 million years, they radiated into the dominant marine predator group of the Cretaceous. The subfamily that matters here is Tylosaurinae, the first mosasaurs to exceed 8 meters. They are named for an elongated bony projection at the tip of the snout, the premaxilla. Tylosaurus means knob lizard. The first Tylosaurus skeleton was described in 1869 by Edward Drinker Cope, from a fragmentary skull shipped to him out of the Niobrara chalk. 3 years later, his rival Othniel Charles Marsh erected the genus name that finally stuck. The 2 men spent the next 2 decades naming and renaming the marine reptiles of Kansas as part of the Bone Wars. The Niobrara chalk was the battleground. The genus Tylosaurus is what survived. By the early 20th century, the species Tylosaurus proriger was reasonably well understood. Adults reached 8 to 9 meters. The largest articulated specimens pushed past 12. And then, in the summer of 1911, a curator at the University of Kansas named Charles D. Bunker walked into a chalk outcrop in Wallace County, collecting birds and mammals, and ran into a Tylosaurus skull almost 6 feet long, eroding straight out of the rock. Bunker was not a marine reptile specialist. He packed up every piece anyway. The skeleton shipped back to Lawrence and went into storage. His original field notes were misplaced. The bones sat unmounted for 88 years. In 1999, the field notes were rediscovered in the KU archives. A paleontology firm called Tribold restored the skeleton and produced the full mount that hangs in the lobby today. The Kansas legislature designated Tylosaurus the official state marine fossil in 2014. The Bunker mount is the reason. That animal in life was probably an adult male, roughly 14 meters long, 8 tons, the largest complete mosasaur skeleton ever recovered on the continent. And in every textbook printed before 2026, that label was wrong. To understand why the reclassification matters, the diet has to come first. What Tylosaurus ate is not a guess. In the summer of 1918, a paleontologist named Charles Sternberg was working a chalk outcrop near Twin Butte Creek in Logan County, Kansas. He pulled out a Tylosaurus skeleton roughly 9 meters long. The skull alone measured almost 5 feet. The specimen went to the United States National Museum, which became the Smithsonian. But inside the Tylosaurus ribcage, packed where the stomach had been, Sternberg found another set of bones. Smaller, partially dissolved, the surfaces pitted as though they had been left in a weak acid bath. They were not Tylosaurus bones, they belonged to a plesiosaur. Sternberg reported the discovery at the Kansas Academy of Science the following year, in a paper short enough to disappear from the literature for the better part of a century. It was not until 2001 that a Kansas paleontologist named Michael Everhart, working at the Sternberg Museum at Fort Hays State University, went back to the original notes. Everhart republished the discovery in 2004 in a journal called The Mosasaur. The conclusion held a 9-meter Tylosaurus weighing roughly 1,100 kilograms had swallowed a 2.5-meter plesiosaur whole, then died with the prey still in its body cavity. The Sternberg specimen is not the only one. A skeleton catalogued as SDSM 10439, recovered from the Pierre Shale in South Dakota, contains stomach contents from at least 3 different prey animals in a single individual Tylosaurus, a smaller mosasaur around 2 meters long, a flightless diving bird called Hesperornis, a bony fish in the genus Benenogmius, possibly a shark, all preserved together inside one rib cage, a sea turtle in the Protostega family, displayed in the same hall as Bunker at KU, carries approximately 100 small puncture marks across its shell and limb bones. The marks match the tooth spacing of a Tylosaurus the size of Bunker. None of the bites show evidence of healing. The attack was fatal. The picture from those specimens is consistent. Tylosaurus ate fish, sharks, sea turtles, plesiosaurs, flightless birds, and other mosasaurs. It was a generalist apex predator that could take prey nearly as long as itself. The problem is that stomach contents are rare. Across the global Tylosaurus record, fewer than 10 well-documented stomach content specimens exist. To build a complete picture, paleontology needs other methods. The first of those methods is teeth. A paleontologist named Femke Holverda, working at the Royal Tyrrell Museum of Paleontology in Alberta and the Naturalis Biodiversity Center in the Netherlands, has spent several years applying 3-dimensional dental microwear texture analysis to mosasaur teeth. The method is straightforward. Different prey types leave different patterns of micro-scratches and micro-pits on tooth enamel. A scanning microscope at high resolution quantifies those patterns. Fish leave one signature, hard-shelled invertebrates leave another, tough vertebrate prey leaves a third. Holverda's team published the first 3-dimensional mosasaur microwear study in Scientific Reports in 2023, working with type Maastrichtian material from the Netherlands and Belgium. They extended the method to the Campanian Bear Paw Formation of Alberta in the journal Diversity in 2025. The results were not what a strict niche partitioning model would predict. The species in the sample did not show clean dietary boundaries, they overlapped, different mosasaurs shared prey, and the differences between species were matters of degree. The second method is engineering. In April 2026, a paper appeared in the journal Paleontology from the Eddy Lab at the University of Liège in Belgium. The lead author was Francesco Della Giustina. The team surface scanned the skulls and jaws of 16 late Cretaceous marine reptile species from North America, 13 mosasaurs, 3 polycotyledid plesiosaurs. For each scan, they reconstructed jaw musculature from attachment scars on the bone, converted the skull into a digital mesh of up to 10 million elements, and simulated biting. The technique is called finite element analysis. It comes from structural engineering. Apply a digital bite, measure where stress concentrates, how efficiently muscle force translates to closing force at the tooth tip, how much energy the skull absorbs before something fails. The Della Giustina team ran every species in the dataset through the simulation. Plesiosaurs, with their long, narrow skulls, came out optimized for fast, low-force strikes. Several smaller mosasaurs clustered with them. The big mosasaurs did not. Tylosaurus proriger and Mosasaurus hoffmanni, the 2 largest taxa in the sample, both around or above 10 meters, sat at the top for raw bite force. They also showed the best ratio of mechanical efficiency to stress, meaning the skulls delivered the most force per unit of muscle effort while absorbing that force with the least risk of fracture. That last part matters. A skull that produces a powerful bite at high internal stress is a skull that breaks. Tylosaurus proriger did not. Its coronoid process, the bony attachment for the main jaw-closing muscle, was lengthened relative to most other squamates, increasing mechanical advantage. The lower jaw distributed stress across the bone, rather than concentrating it at the joint. The engineering supports what the stomach contents already showed. The animal could close on a plesiosaur and survive the experience. 3 independent methods, stomach contents, dental microwear, finite element modeling, they converged on the same answer. And then, in the spring of 2026, that answer changed shape. Amelia Zietlow began the work as a doctoral student at the Richard Gilder Graduate School at the American Museum of Natural History. She found a mosasaur skeleton in the museum research collection cataloged as Tylosaurus proriger. The teeth were too finely serrated, a trait uncommon in mosasaurs. The bones were too large. She compared the specimen to Cope's original proriger holotype at the Harvard Museum of Comparative Zoology, the one described in 1869. The differences were consistent. With her co-authors Michael Polson of Southern Methodist University and Ronald Tukoski of the Perot Museum of Nature and Science in Dallas, Zietlow widened the search. They found more than a dozen specimens at multiple institutions sharing the same unusual features. Finely serrated teeth, a deep depression in the back of the skull called the nuchal fossa, where the powerful neck muscles attached, a distinctive three-lobed quadrate joint at the back of the jaw. 12 specimens in total that the team reclassified. The paper landed in the Bulletin of the American Museum of Natural History in May 2026, issue number 482. The new species name is Tylosaurus rex. The Latin translates as Tyrant King of the Knob Lizards. The holotype, PMNS 8029, sits at the Perot Museum in Dallas. It was excavated in 1979 near a reservoir called Lake Ray Hubbard. For decades, it was known informally as the Heath mosasaur. Two of the other reclassified specimens are worth saying out loud. One is a skeleton called Sophie, on display at the Yale Peabody Museum in New Haven, long considered one of the best preserved Tylosaurus skulls in the world. The other is Bunker, the mount at the University of Kansas, the state marine fossil, 8 tons in life. It is not Tylosaurus proriger, it is Tylosaurus rex. The reclassification is not just a name change. The Zietlow paper rebuilt the dataset used to test mosasaur evolutionary relationships, which had remained largely unchanged for almost 30 years. Within the new analysis, Tylosaurus rex sits as a younger sister species to Tylosaurus proriger. The Kansas proriger material is around 84 million years old. The Texas rex material is around 80 million. 4 million years separate them. The Bunker specimen, on the older end of the rex range, represents the northern extension of the species into what is now Kansas. There is one more detail. A second Texas specimen, nicknamed the Black Knight, also at the Perot, is missing the tip of its snout. Its lower jaw is fractured. The damage, the team argues, could only have been caused by another animal of its own species. Tylosaurus rex was not just biting plesiosaurs and sharks. The largest predators in that sea were biting each other. Here is where the story lands. 80 million years ago, 2 unrelated apex predators were carrying the same name. One walked on land in Laramidia and is called Tyrannosaurus rex. The other swam in the Western Interior Seaway and, as of the 2026 redescription, is called Tylosaurus rex. They were separated by an inland ocean. They never met. Evolution produced 2 T-Rexes in the same late Cretaceous world, one terrestrial, one marine, and the marine version sat unrecognized in American museum drawers for more than a century. And there is a broader pattern in that. Marine apex predators are systematically underdescribed because the field effort that finds them is much smaller than the field effort that finds dinosaurs. Excavations concentrate on the terrestrial formations that produce hadrosaurs and tyrannosaurs. Marine reptile collections, especially older ones, are reanalyzed only when a curator like Zietlow opens a drawer and starts measuring. The Bunker skeleton was collected in 1911. Its field notes were lost for 88 years. Then it was misidentified for another 25. The largest mosasaur in North America was sitting in plain sight that entire time. The Zietlow paper ends on a note that applies to every museum collection on the continent. The taxonomic infrastructure for mosasaurs was running on a data set that could not resolve the species the team has now described. Other specimens, in other drawers, are almost certainly waiting for the same treatment. For now, the Western Interior Seaway has its king, the Smoky Hill Chalk of western Kansas, where Sternberg found a plesiosaur inside a Tylosaurus in 1918, where Bunker pulled an 8-ton skeleton out of an eroding outcrop in 1911, is the ground where the question of what hunted that ocean was first asked. The answer, 115 years later, is Tylosaurus rex. Till the next find, this is Bone Atlas.